Hi, welcome back to my channel. In this video, I will be talking about the beta oxidation of fatty acids in an easy way. So first of all, what exactly is beta oxidation of fatty acids? So if you think about the word beta oxidation, there are many other oxidations as well, but beta oxidation is the most common. And basically, in this, the two carbon atom at a time are cleaved from carboxyl end of activated fatty acid as acetyl CoA. Now you will understand this once I explain the pathway. So site is liver, adipose tissue, muscle. Organelle, most common organelle is mitochondria, in which beta oxidation fatty acid occurs. Let's talk about the pathway. So first of all, you should know that first we have there are three stages of the pathway. So first we have the activation, then we have transportation, and then we have oxidation, then hydration, then oxidation. All right, then cleavage. Okay, so let's just understand the pathway. The pathway is rather easier to understand. So first of all, I use black color first. So we have we'll start from fatty acid. Okay, now this fatty acid is converted into acyl CoA through the enzyme thiokinase, which is also called as acyl CoA synthetase. Okay, now this problem this is present in the cytosol. This reaction is occurring in the cytosol, but I told you that beta oxidation occurs in mitochondria, so we need to go to the mitochondria. Now this occurs through some carnitine derivatives. So carnitine, now in this what happens is you have to remember one liner here, that short and medium chain fatty acids do not require carnitine derivatives. Okay, now through this carnitine, the acyl CoA finally reaches into the mitochondria. Okay, now I told you there are three steps. So this is first step done, which is activation, and it utilizes 2 ATP. Okay, remember this. Then this acyl CoA is now in mitochondria. See, now second step I told you was transportation. So this is transport, this is also done. Now third step I told you is oxidation, then hydration, then oxidation, and then cleavage. Okay, now let's just go through the steps. So now acyl CoA is converted into trans-enoyl CoA by the action of enzyme acyl CoA dehydrogenase. This is oxidation. Then hydration, so hydratase, and we have beta hydroxy. We will add just the OH group, right, on beta position, acyl CoA. Okay, then again I told you hydration, and then oxidation, again we have dehydrogenase enzyme, so it will form beta keto acyl CoA. This beta acyl CoA, keto acyl CoA, now is the time for cleavage. So it will be converted into acetyl CoA by the action of thiolase. Now mind you, it is not thiokinase, it is thiolase. Okay, and here we also have acyl CoA. Now this acyl CoA will be produced with less than 2 carbon, minus 2 carbon, from which it was initially having. So if you have palmitic acid, suppose 16, then 14 will be left. Why so? Because this 2 carbon atom is given to the acetyl CoA. Now see this again, 2 carbon atom at a time are cleave to form carboxyl end of from the carboxyl end of the activated fatty acid as acetyl CoA. Okay, so this is what I was telling in the intro. So if over here you have to know that here we have FeDH2 produced, and here we have NADH. Okay, now so let's talk about the energetics. So this is one cycle. Okay, so just give me a second. here we'll also talk about energetics. So let's just understand the energetics. So to remember this, for even carbon atoms, the number of cycle, this you have to by heart, is n by 2 minus a, and the number of acetyl CoA formed will be n by 2. Now let's just take the example of, so first just understand, in one cycle you have one FADH formed and one NADH. So this is 2.5 and this is 1.5 ATP, right? So this is 1.5 ATP and this is 2.5 ATP. So this is like 4 ATP, right? And remember that 2 ATP were used. Okay, so let's just take the example of palmitic acid. So palmitic acid is 16 carbon, right? So let's talk about number of cycle. So n by 2 minus 1, so 16 by 2 minus 1, which is 7 cycles, and number of acetyl CoA formed will be n by 2. So this is 16 by 2, which is 8. Now in energetics, you should know that this acetyl CoA will further go to TCA cycle, and through one cycle it will produce 10 ATP. Okay, so let's talk about the energetics. So energetics, so we have 7 cycles, so this is 7 into 4, which is 28 ATP, plus you have 8 acetyl CoA, which is going to TCA cycle. One TCA cycle has 10 ATP, so 8 into 10, which is 80, so 108 ATP is our this formed. And then this minus 2 ATP utilized in activation, so resultant is 106 ATP. So the answer is 106 ATP. Now all of this is about even. What about odd chain fatty acid? Now this is the question that they usually ask, and very repetitive question. Here in odd chain fatty acids, we have propionyl CoA, which is converted into succinyl CoA. Okay, and you have presence of B12. Okay, so this is it, and we have discussed this. Now regulation. Regulation may you have fed stage and fasting stage. So in fed stage you have increase in insulin and glucagon ratio. There is increase in malonyl CoA, right? So due to that there is decrease in the beta oxidation, whereas in fasting there is increase in beta oxidation. So this is all about beta oxidation of fatty acids. And one more thing, the cut is made between alpha and beta carbon. Okay, so yeah, so this is about beta oxidation of fatty acids. If I made it easier for you to understand, do give it a like, subscribe, share, comment, and yeah, thanks for watching.